All right, so this is the second lecture for Chapter 15, and in this lecture we're going to talk about the regulation of glycolysis.
So again, we come back to our kind of 30,000 foot view diagram here of metabolism. So as you can see, the glucose is at the center of it, glycolysis is at the center of it. So the flux of glycolysis is really carefully controlled to ensure that there's appropriate utilization of glucose by tissues that primarily rely on it. And the examples of that are skeletal muscle, heart muscle, and the brain. And that makes sense because these are critical for life. And really where a lot of this regulation occurs is really in one of two places, is hepatocytes, the liver during especially fasting, and then also in muscle cells during intense exercise. So in terms of fasting and prolonged starvation, as we indicate here over 3 days for prolonged starvation, this is really important because what happens is that the liver switches to using fatty acids instead of glucose, and what that does is it spares glucose to be used by these other tissues, such as the heart, the brain, and because they primarily rely on it. And the liver does just fine breaking down fatty acids from triglycerides, converting them to acetyl-CoA through beta oxidation, and then acetyl-CoA can enter the citric acid cycle and provide plenty of ATP for hepatocytes to function. At the same time, hepatocytes are synthesizing glucose via gluconeogenesis from pyruvate, and the purpose of that is not so that the liver can use that glucose, it's to produce glucose that can be used by peripheral tissues, such as heart muscle, brain, skeletal muscle. So by switching to fatty acids, the liver essentially preserves glucose for other tissues. The serum concentration of glucose is increased after a meal, which is no surprise. And then as a result, the liver rapidly removes that glucose from the blood, it takes it up and then rapidly uses it for glycogen formation, essentially to be storing glucose for these times of fasting, and then also for fatty acid synthesis. So you have increased glucose after a meal. That's going to first go towards glycogen storage and glycogen formation. So once you've maxed that out, then you're going to come down here and form acetyl-CoA, which is then going to be used to synthesize fatty acids, which are going to pair with glycerol and be stored as triglycerides in adipose tissue. The reason why you will eventually shift over here is when you have an overabundance of glucose, you've maxed out these processes here, and they're going to feedback, inhibit, and shift the process of metabolism this way from an energy producing state to a energy storage state, which is where you're storing fatty acids. So really in the fed state, you want to be thinking of energy storage, because from the body's perspective, it doesn't know when another meal is coming. So it's got to be able to store out. You don't want to just blow through all the energy and use it all up, especially if you're just kind of sitting and chilling. Versus the fasting state is energy utilization and really the mobilization of storage, which are essentially glycogen and fatty acids. Also, those are the two main ones, and also in really severe, you can have amino acids as well. Bottom line is that hepatocytes are taking glucose up rapidly, storing it as glycogen. When that's maxed out, then they're storing it as fatty acids in the fed state. So a thing to note is that the concentration of glucose in the blood is equal to the concentration of glucose in the liver, and this is ensured by those first two regulatory steps of glycolysis. So we talked about this. First, you have this GLUT2 transporter, which allows glucose to be readily taken up into the cell. This GLUT2 transporter has a high Km, which means that it has a low affinity, and what that does is it ensures that it's not saturated by physiological concentrations of glucose in the portal circulation that are coming from the GI tract, because remember, the GI tract absorbs it, then you go to the portal vein, which is then going to go bring everything to the liver. So glucose is absorbed here, travels to the portal vein, and then is taken up by the liver. And so when you have high amounts of glucose, you don't want to have any of it sitting out here. You want to make sure that it's getting taken up very efficiently and rapidly by hepatocytes. The other thing is that glucose uptake into hepatocytes is independent of insulin. So there's in peripheral tissues, remember, and this is what eventually causes different forms of diabetes, is where peripheral tissues can't take up glucose in response to insulin. In hepatocytes, insulin has no impact here. It's not dependent on insulin. So as you increase the glucose concentration, you're going to be able to continually uptake glucose into the hepatocytes, and it's not waiting on an insulin signal. Second level of regulation here is the glucose phosphorylation. So again, glucose gets taken up into the cell. First thing that happens is you invest in ATP into phosphorylating glucose to form glucose 6-phosphate, key intermediate. Remember, adding that phosphate group on to glucose at this 6 position prevents glucose from traveling back outside of the hepatocyte. So it contains it into the cells so that we can go to work on it and either do glycolysis, glycogen formation, or eventually fatty acids production. Now, you have 2 enzymes that carry this out. You have hexokinase and glucokinase. Hexokinase is in every single cell in the body. Glucokinase, however, is only exclusive to hepatocytes and beta cells of the pancreas. Big differences between glucokinase and hexokinase is glucokinase is not subject to feedback inhibition. So for hexokinase, this can come back in negative feedback. So once you've reached a certain tipping point, this will come back and inhibit it. Versus glucokinase, you don't want to have any limit there because you want to be taking up as much glucose as possible during the fed state so that you can max out those glycogen storages and then eventually, from the body's perspective, provide as much storage as possible by producing fatty acids as well. The other thing is, so in the fed state, you're going to have increased glucose in the blood, then you're going to have increased insulin in response to that, and then insulin is going to stimulate synthesis of glucokinase, which makes sense because you're going to have a higher level of glucose. You want a higher capacity, enzyme capacity, to be able to process this glucose coming into hepatocytes. On the flip side, in the fasting state, you have decreased glucose, and you're going to have an increase of a hormone called glucagon, which essentially helps stimulate that gluconeogenesis and glucose synthesis. And we'll talk more about that in the gluconeogenesis lecture. And that's, again, to produce glucose to be used by peripheral tissues. So when you have increased glucagon during the fasting state, you're going to actually decrease synthesis of glucokinase. So point is that the levels of glucokinase are sensitive to what state the body is in and then what the levels are of these hormones. Glucokinase has a lower affinity for glucose. This is an arrow. It should be an up arrow. So it has a higher Km for glucose, but it has a higher metabolic capacity compared to hexokinase. That makes sense. It's so that it doesn't get saturated again by these concentrations of glucose. It allows it to have a much higher capacity to handle these high levels of glucose that come in during the fed state. When glucokinase is mutated, it's associated with maturity onset diabetes of the young, MODY. It's just a nice little nugget we threw in there for you. Be aware for either a biochem exam or a board exam could be asked that. The liver also has hexokinase, as I mentioned, which is present in all other cells. But again, even in the liver, hexokinase is subject to that feedback inhibition from glucose 6-phosphate. It's also saturated by normal concentrations of glucose present in hepatocytes. So hexokinase gets saturated by these high levels of glucose during the fed state, but like I was saying, glucokinase does not get saturated by those. And so that's why glucokinase, again, has a higher metabolic capacity. And then another thing I'll point out here is that when you have an overabundance of glucose, this also explains why overabundance of carbohydrates leads to increased fatty acids, increased adipose tissue, and then eventually weight gain. All right, so given that phosphofructokinase 1 or PFK1 is both the rate-limiting step and the committed step of glycolysis, it's going to be subject to several levels of regulation for that reason. And really, there's 2 categories of regulation, if you will. The one that we'll talk about in this slide is energy metabolism, and then the second one, which we'll talk about on subsequent slides, is hormonal control. Different hormones are released in response to really whether the body's in the fed or the fasting state, and then those hormones influence the flux of glycolysis and thus energy production. And again, we'll talk about that in subsequent slides. So for energy metabolism, like I said, PFK1 is sensitive to the energy state of the cell, and specifically, it's sensitive to the levels of ATP and citrate. That makes sense. The ATP is, obviously the energy source that cells use, and then citrate is a key intermediate or the first intermediate for citric acid cycle. ATP is a negative allosteric regulator of PFK1, while AMP is a positive allosteric regulator. And we indicate that in the diagram. So things in green are activators, and then things in red are inhibitors. So AMP is an activator, and then ATP and citrate are inhibitors. And these are really just, again, indicators of the energy state of the cell. So in a low energy state, you're going to have increased levels of AMP. That's going to stimulate PFK1 because you are in a low energy state. So you want to increase the flux of glycolysis, increase ATP production, and change that energy state. Versus when you're in a high energy state, you're going to have increased levels of citrate and ATP. And so you're going to want to be able to spare oxidation of glucose via glycolysis. And the way you would have high energy state is due to the metabolism of other metabolic fuels, such as fatty acids. So if you're getting enough energy production from fatty acids, this particularly can happen in the liver. Glucose oxidation can be spared, and so you'll inhibit this, and then you can force the path of glucose to more follow, say, glycogen formation. So you have enough energy around, you have this abundance of glucose, you're going to force it into glycogen synthesis because you want to store it essentially for a fasting period where you won't have glucose, and then you can break glycogen down and have more glucose in the fasting state. Citrate is the first intermediate of the citric acid cycle, and like we said, it's a negative allosteric regulator. And so what that does, as we indicate in this diagram here, is it directly links citric acid cycle activity and really what's going on in the mitochondria here to the flux and rate of glycolysis. So we have PFK1 here as the main enzyme. So if you have increased flux of citric acid cycle, you're going to have increased levels of citrate. So citrate will accumulate in the cell, and then it'll come back and negative feedback inhibit on PFK1. And so again, that directly links the activity of the citric acid cycle to the flux and rate of glycolysis. And again, this is going to be during high energy states. And so you want to be able to essentially spare glucose for things such as, synthesizing glycogen and storing glucose for a fasting period. All right, so now for the second category, hormonal control. Hormonal control primarily affects the rate of glycolysis in the liver. And remember, the liver is essentially a central hub for metabolism and especially carbohydrate metabolism. And so it helps coordinate the generation of metabolic energy from hepatic glycogen, which is a huge storage center for glycogen, fatty acids in adipose tissue, and then also glucose in skeletal muscle. So again, these hormones are being secreted in response to whether you're in the fasting state or the fed state, and they're helping coordinate these different processes, especially in the liver and then also in adipose tissue and then in skeletal muscle as well. So during the fasting state, you're going to have increased levels of glucagon, which is then going to bind and stimulate its receptor. And then the receptor, remember, it's a G protein coupled receptor. We talked about these in Unit 2. It's going to activate the enzyme adenylate cyclase, which is going to take ATP and convert it into cyclic AMP. And then cyclic AMP is going to activate protein kinase A, which will then phosphorylate several proteins involved in metabolic control. Remember, we talked about in hepatocytes, it's actually going to decrease synthesis of glucokinase because glucokinase, remember, is involved in periods of fasting. And so that's just one of the effects of protein kinase A in hepatocytes during the fasting state. Now, what's important to point out is that pKa does not directly act on PFK1. However, it does definitely affect the flux and activity of PFK1. So in the liver specifically, pKa does phosphorylate an enzyme called fructose 6-phosphokinase /fructose 2,6-bisphosphatase, which is also known as phosphofructokinase 2, abbreviated to PFK2. Now, PFK2, it will come down here, is a bifunctional enzyme. So it has a kinase activity when it's going in one direction, and it also has phosphatase activity. So it does both. And so when you're going in this direction, this is the kinase activity, you're going to phosphorylate fructose 6-phosphate using an investment here of ATP, and you're going to phosphorylate on this 2 position to give you fructose 2,6-bisphosphate. Now, if you do the phosphatase activity in the reverse, you're going to go this way, and you're going to take this phosphate off of this 2 position and regenerate fructose 6-phosphate plus an inorganic phosphate. So in the previous lecture, we were going through the steps of glycolysis. You may have been wondering, as I was wondering when I first learned about glycolysis, is what's the point of diverging right here, right before you're going to enter this committed and rate limiting step, and using up an ATP to form this intermediate fructose 2,6-bisphosphate? Well, for one thing, fructose 2,6-bisphosphate, when it's formed, is actually a positive allosteric regulator of PFK1. Now, you may be saying, so what? What's the point of making this to help boost this enzyme? You got to use up an ATP in the process. You know, the cell has an abundance of ATP. And really, you can essentially afford, the way I say this is as an investment, you can afford this investment of this ATP because you just have enough of it. You can think of it as like a small donation or a small investment of the cell for its own good. So you're investing here in producing some fructose 2,6-bisphosphate. And what it does is by increasing this, you help further along the process of glycolysis. So it's really, again, you may still be kind of at a loss of why you still do this, but it's you want to think of it as it's a small investment to help further the overall energy state of the cell. Because although you use up this one ATP here, by furthering glycolysis, you further pyruvate production, and then you eventually further citric acid cycle flux and ATP production in the mitochondria, which generates numerous ATP, as we've gone over many times now. By investing in this, it's in this overall cell's energy state's best interest. So, to summarize here, if you increase the kinase activity of PFK2, you'll increase levels of fructose 2,6-bisphosphate, which will then come over here and activate PFK K1 and increase activity of that, thus increasing eventual ATP production. On the flip side, if you're gonna go this way, you're going to decrease levels of fructose 2,6-bisphosphate, and then that's going to decrease PFK1 activity, decreasing glycolysis. So an important thing I'll point out here is that phosphorylation of PFK2 induces the phosphatase function. So if you have PFK2 where it's not phosphorylated, it's acting as a kinase. Then if you come along, add a phosphate to it, and you attach this phosphate, now you have phosphatase activity. And then again, if you revert back to where you release this phosphate, you enhance kinase activity. And then the other thing to point out here is that cyclic AMP actually directly inhibits the kinase activity. So cyclic AMP and protein kinase A kind of have this double effect of enhancing phosphatase activity, because if you increase pKa, you're going to increase phosphorylation of PFK2, thus enhancing its phosphatase activity. So during the fasting state, you have decreased serum glucose. This is during fasting. Increased glucagon, which again is the source of all this. And you're going to have, like I said, increased cyclic AMP, and this is going to lead to increased pKa. And then that's going to, as I've explained here, going to decrease the kinase activity and increase the phosphatase activity of PFK2. So what that's going to do is then shift this reaction this way, because you have the phosphatase activity, because again, you have increased levels of both of these. Decrease the level of fructose 2,6-bisphosphate, which will then decrease the level of PFK1 activity, which will then decrease glycolysis. And on the flip side, which is glucagon's intent, is going to increase glucose synthesis, which is part of gluconeogenesis, which is de novo glucose synthesis. And that makes sense. I mean, during the fasting state, you don't want to be breaking down glucose in the liver. Specifically, the liver uses fatty acids during the fasting state to help spare glucose for peripheral tissues that more heavily rely on it, such as the brain, red blood cells, heart muscle, tissues like that nature that are critical for life. In that state, this helps the hepatocytes carry out its function by shifting more towards glucose synthesis, and again, helping supply glucose for peripheral tissues. So again, this step here, while it at first may seem pointless, also serves an important step, not only during helping increase the flux of glycolysis during the fed state, but during the fasting state, it also plays a critical role, because then it also helps dampen down this activity of PFK1. It further helps decrease the activity of glycolysis and further increase the activity of gluconeogenesis to help further the end goal of producing glucose for peripheral tissues. All right, so in the muscle, there's a key distinction to make in terms of hormonal control of glycolysis. And that really is that protein kinase A does not phosphorylate PFK2. So cyclic AMP and protein kinase A do not affect the rate of glycolysis in the muscle. And you'll see why that's important in a second. So again, just to put some labels in here, PFK2 would be here, which would be producing fructose 2,6-bisphosphate when it's in the kinase form. When it's in the phosphatase form, converts this back to fructose 6-phosphate. And then you have PFK1 here, which is again that committed rate limiting step for glycolysis. The reason this is important is because remember, skeletal muscle does not synthesize glucose during fasting. It doesn't synthesize glucose at all. And the reason that's important is because remember, in the liver, you want to decrease glycolysis to allow the liver to then carry out gluconeogenesis and produce glucose. Glycolysis and gluconeogenesis are two opposite processes. One is the breakdown of glucose to produce energy. The other one is the synthesis of glucose from various precursors in the cell. You can't carry them out simultaneously. It's either one or the other. And so in the muscle, you can't carry out gluconeogenesis, so it doesn't matter. There's no advantage to having protein kinase A and cyclic AMP inhibit the kinase function. And so, because remember, when the glucagon is high during fasting in the liver, cyclic AMP and protein kinase A decrease kinase activity and enhance phosphatase activity, which would drive this back towards here, decreasing the concentration of this, which would then decrease the stimulation of PFK1 and decrease the flux of glycolysis. Remember, that's in the liver. In the muscle, you don't need to do this because you're not carrying out gluconeogenesis. In fact, you're doing quite the opposite. You're breaking down glycogen stores during fasting in the muscle to then allow you to carry out glycolysis. So during fasting, you need to carry out glycolysis to produce ATP eventually. So there's no advantage to decreasing fructose 2,6-bisphosphate. In fact, it's actually advantageous, and this is what happens, is this reaction moves towards producing fructose 2,6-bisphosphate, which then stimulates PFK1 and then stimulates glycolysis. And so again, as we say here, during fasting, glycogen in the muscle provides the glucose that can be directly broken down via glycolysis. And what's nice then is you don't have any interference from cyclic AMP and protein kinase A, which are stimulated by glucagon during fasting. And so during fasting, even though glucagon's at work here, you're breaking down glycogen, carrying out glycolysis, and then you don't have any interference where you're preventing any increase in concentration of this, which would also further your ability to carry out glycolysis. Again, that all comes back to the very key fact that you do not synthesize glucose in the muscle. Now, in the liver, glycolysis is inhibited during fasting versus the muscle. So here, we'll do during a fast, in the liver, you're inhibiting glycolysis. We'll say that's why that's important in a second. And then in the muscle, you're stimulating glycolysis because the main source of energy is going to be glycogen breakdown. However, during fasting in the liver, you want to be synthesizing glucose and breaking down glycogen so that you can provide glucose for other tissues that really rely on them. Remember, that's the whole key. The liver almost is selfless during fasting, where it gives up its glucose for other tissues such as the brain and red blood cells that really need them. And so the two main sources of glucose during fasting from the liver are glycogen stores. So you break down glycogen to provide glucose for peripheral tissues. And remember, the liver doesn't use that glucose broken down from glycogen. It shoots it out into the bloodstream so that it can be used by peripheral tissues. And then gluconeogenesis. And we'll talk about glycogen metabolism and gluconeogenesis in much more detail in their respective lectures, but it's important to reference it here because it helps you further understand the regulation of glycolysis and why you would decrease the flux of glycolysis in the liver versus why you don't decrease it in the muscle. Key concepts to understand here. Now, what enables the liver to do this is during fasting, it uses fatty acids as its metabolic fuel. And remember from our main diagram for metabolism, breakdown of fatty acids gives you a number of acetyl-CoA, which can then go into the citric acid cycle and via the electron transport chain produce plenty of ATP to be used by the liver while it's producing glucose for other tissues. And so again, just to come back to this concept here again, cyclic AMP and protein kinase A, they're going to shift this reaction more towards this way, decreasing fructose 2,6 bisphosphate, decreasing PFK1 activity, which then decreases glycolysis, but in turn increases gluconeogenesis. And you also have glycogen breakdown, but again, since you have decreased flux of glycolysis, that glycogen, instead of being shunted towards oxidation via glycolysis, it's going to be shunted out into the bloodstream. So another level of regulation of glycolysis, especially in the liver, that we'll talk about briefly here on this slide, is the sensitivity of the enzyme of glyceraldehyde 3-phosphate dehydrogenase to the NADH-NAD ratio within the cytosol. So this ratio regulates the activity of this enzyme, which also contributes to the overall flux of glycolysis. And so just to remind you, you have PFK1 here, which takes fructose 6-phosphate and converts it into fructose 1,6 bisphosphate, which is a 6-carbon molecule. Then you split that molecule into 2 3-carbon molecules. So this is a 3-carbon molecule, this is a 3-carbon molecule. If you remember from the previous lecture, this gets rapidly converted to glyceraldehyde 3-phosphate, and that's what gives you 2 equivalents of this. Now, to blow up this reaction here, we show it here. Here's glyceraldehyde 3-phosphate. This is catalyzed by glyceraldehyde 3-phosphate dehydrogenase, and this produces NADH. So this NADH to NAD ratio regulates this enzyme's activity. And actually, when you have increased levels of NADH, it decreases the activity of the enzyme, as we indicate here. Now, one thing to note here, and we'll talk about this more in fatty acid metabolism, but high levels of NADH relative to NAD+ in the liver indicate high levels of fatty acid oxidation. And that's because the reactions that carry out fatty acid oxidation, which is going to be increased during fasting, this is going to lead to increased levels of NADH, again, which will indicate to the liver that the body is in a fasting state and it needs to begin the process of switching from using glucose to using fatty acids so that then it can carry out gluconeogenesis and then synthesize glucose and break down glycogen to provide glucose for peripheral tissues. Now, as a result of decreasing the activity of this enzyme, glyceraldehyde 3-phosphate, this actually contributes to reducing the rate of glycolysis in part. So it's one of the contributors. You affect it up here by directly acting on PFK1. You also affect it with levels of fructose 2,6 bisphosphate. And then here's another area where you affect the overall flux of glycolysis at glyceraldehyde 3-phosphate dehydrogenase. So it's another way of reducing the flux of glycolysis. This mechanism of NADH relative to NAD also is responsible for hypoglycemia that occurs after heavy alcohol consumption. We'll talk about this in more detail in chapter 30, the alcohol chapter, in unit 4, the nutrition unit. Basically, the things to take away at this point is that alcohol metabolism, when it gets broken down, the two enzymes that break it down actually produce high levels of NADH. NADH then gets used up by essentially 2 reactions. This reaction right here, where pyruvate gets converted to lactate. Also, heavy alcohol consumption results in increased lactate, which is, again, lactic acidosis, or potentially can produce lactic acidosis. So it uses up NADH that way because it's an effort by the cell to balance out this ratio of NADH to NAD+, because you need NAD+ to carry out critical reactions. There's another reaction where you have oxyl acetate converted to malate, and that uses up NADH as well. By using up NADH to convert pyruvate to lactate, you decrease the level of pyruvate. And pyruvate, as we'll talk about in more detail in the gluconeogenesis lecture, is one of the critical starting points for gluconeogenesis because it's the reverse of glycolysis. You're taking pyruvate and through a series of steps converting it back to glucose. And so if you don't even have pyruvate to start out with, that's going to lead to an inability to effectively produce glucose, which results in hypoglycemia. Again, we'll get into this in more detail in the gluconeogenesis lecture. and in the alcohol lecture as well in Unit 4. But just to give you a sense of how this all ties together and why this NADH to NAD plus ratio is so critical. All right, so another layer of regulation we'll talk about is at the enzyme called pyruvate kinase. Now there's two forms of pyruvate kinase, PKL, which is the liver isozyme, which we'll talk about in this slide, and then there's PKM for muscle, which we'll talk about in the next slide. There's key distinctions to make between the two. So the liver form is inhibited when phosphorylated by protein kinase A, which if you remember, you have increased levels of that during fasting, which is stimulated by increased levels of glucagon, which also has increased levels of cyclic AMP. And we indicate that down here. So cyclic AMP, as we indicate in here in red, inhibits pyruvate kinase, which again carries out the conversion of phosphoenol pyruvate to pyruvate. This is a step that generates an ATP, so it's an important step, both for that reason and for the reasons we're talking about on this slide, where it's highly regulated during fasting especially. So you have increased levels of cyclic AMP. The other thing, we'll add in protein kinase A here, that this also will inhibit pyruvate kinase. The other thing here is that ATP and alanine are also allosteric negative regulators. And so when you have high levels of these, high ATP, high alanine, those are really indicative of a high energy state. So an increased energy state. And so during fasting, the way you get to that high energy state in the liver is that you try to preserve glucose by conducting high levels of fatty acid oxidation. And remember, the reason why you're carrying out high levels of fatty acid oxidation is so that you can synthesize glucose to be used by peripheral tissues, such as the brain, red blood cells, so on. So you use fatty acid oxidation to help produce ATP so that the hepatocytes can have a high energy state as well. So a thing to point out here is during fasting, so during a fast or starvation, you have a number of things that are going on. So in the liver, you're going to have increased fatty acid oxidation. What does that result in? High levels of ATP. And ATP is going to inhibit pyruvate kinase, which will decrease glycolysis. And why are you trying to decrease glycolysis in the fasting state in the liver? Is so that you can increase the capacity to carry out gluconeogenesis, which will synthesize glucose for tissues, peripheral tissues. So because remember, these two processes, they can't act simultaneously. It's one or the other. So if you're having decreased glycolysis, then you can have increased gluconeogenesis. If you're carrying out glycolysis, you're not going to have high levels of gluconeogenesis. The other thing that's happening in the body during fasting, in the muscle, you're having breakdown of the amino acid alanine. So alanine is part of the protein structures, and then you're breaking it down to form alanine, which is then shipped off to the liver. And we'll talk about this process in more detail in the amino acid metabolism lectures. But for this purposes, you just want to know that you have high levels of alanine in hepatocytes. It got there from the muscle. That's what's important to know at this point. And that what's really important is that alanine inhibits pyruvate kinase. Now, why are they shipping alanine from the muscle to hepatocytes? Well, alanine is actually used in gluconeogenesis. In fact, alanine, we'll draw it down here so that's not too crowded, is used to synthesize pyruvate. So that's why you're producing high levels of alanine by breaking it down from the muscle and shipping it to the liver, as you're using it to synthesize pyruvate to be used in gluconeogenesis. And then the last thing is that as we've indicated, is you have high levels of glucagon. And so that's going to increase levels of cyclic AMP, which inhibit pyruvate kinase, which also inhibits levels of protein kinase A, which also will phosphorylate pyruvate kinase and decrease that as well. So again, the three things to take away from this slide: increased fatty acid oxidation produces high levels of ATP for hepatocytes so they can function. In turn, that inhibits pyruvate kinase. You're also carrying out gluconeogenesis. Alanine is one of the precursors you use to produce pyruvate. That alanine is coming from the muscle. And then at the same time during fasting, you have high levels of glucagon. Which is resulting in high levels of cyclic AMP, high levels of protein kinase A, which are both decreasing activity of pyruvate kinase, which again, all of three of these are resulting in decreased glycolysis so that you have increased capacity for gluconeogenesis to synthesize glucose for peripheral tissues. Now, in the muscle here, pyruvate kinase is not affected by protein kinase A phosphorylation or alanine. And the reason why it's not affected by alanine is, for one thing, you're breaking down alanine in the muscle to ship it off to the liver for gluconeogenesis. And the reason why it's not staying in the muscle is because the muscle does not carry out gluconeogenesis. It does not produce glucose. So there's no reason for it to be useful here in the muscle, and there's no reason for it to affect the activity level of pyruvate kinase. And remember, the other thing is that in the muscle versus the liver, in the muscle, you're increasing levels of glycolysis because you're not relying on fatty acid oxidation, because in the muscle, you're not trying to preserve glucose for other tissues. The muscle needs glucose. It's its main source, its primary source of energy metabolism. So you have increased levels of glycolysis, you have increased levels of glycogen breakdown, which is then going to go into glycolysis. And so really there's no reason for having alanine or protein kinase A phosphorylation decrease pyruvate kinase flux. In the muscle, you want to have as the highest level of glycolysis flux as possible to help produce energy so that muscle can function in the fasting state. All right, so this is a summary slide that brings together a number of points we've made over the last couple slides and help tie them all together to really bring them into a cohesive story that explains really decreasing glycolysis in the liver during fasting and starvation. Now, remember, the reason why you want to decrease glycolysis is because you want to increase gluconeogenesis. And remember, the reason you want to increase gluconeogenesis is because you want to synthesize glucose for peripheral tissues. And remember, you can't carry out glycolysis and gluconeogenesis at the same time, hence why you have to decrease glycolysis to enable this ability. So if you're not using glycolysis, you need to produce energy from somewhere else. You need to get ATP from somewhere else. And the way the hepatocytes do that is increasing fatty acid oxidation. So that results in high levels of NADH. And high levels of NADH result in really two key effects on regulating glycolysis. First, you increase levels of ATP because through high levels of NADH, and then also through fatty acid oxidation producing acetyl-CoA, which enters the citric acid cycle and produces high levels of ATP. ATP comes and feedback inhibits on two places. Phosphofructokinase 1, which again is that rate limiting and committed step, so PFK1, and then down here at pyruvate kinase. So both of these places are sensitive to energy-rich state, as we've mentioned. Because although you're not using glucose, you're still in a high energy state because you have high levels of ATP from fatty acid oxidation. So those are two effects right there. The other effect is by having increased levels of NADH, you affect this enzyme, glyceraldehyde 3-phosphate dehydrogenase, which remember down here, we'll indicate it here, carries out this reaction of converting glyceraldehyde 3-phosphate into 1,3-diphosphoglycerate, which yields an NADH. Now remember, this enzyme is sensitive to high levels of NADH. And so by increasing fatty acid oxidation, you're producing high levels of NADH in the cell, which will in turn affect the activity of this enzyme. The other thing to point out is that, as we've mentioned, is that during the fasting state, glucagon is released from the alpha cells of the pancreas into the blood, and that results in high levels of cyclic AMP and protein kinase. Remember, a few slides ago, we mentioned one of the effects of that is decreasing levels of glucokinase. Remember, that's important because glucokinase catalyzes the investment of this ATP to phosphorylate glucose and converting it into glucose 6-phosphate, which, remember, this phosphate attached to it prevents glucose from leaving the cell and going back out into the bloodstream. Now, if you're trying to carry out glycolysis, that's important. However, during fasting, you're not trying to carry out glycolysis, and in fact, you're actually trying to take whatever glucose you have in the cell, in the hepatocytes, and release it back into the bloodstream to be used by peripheral tissues. So you don't want to phosphorylate glucose. You don't want to contain it within the cell. You want to be able to release it. So you don't need high levels of glucokinase. So glucagon leads to decreased levels of glucokinase. The other thing that high levels of cyclic AMP and high levels of protein kinase, because remember this activates this, is that at first it increases the phosphatase activity of PFK2. Now remember, PFK2 is this bidirectional enzyme here that has kinase function that can produce fructose 2,6-bisphosphate, and then it has phosphatase action, which in this case you're increasing phosphatase activity, which brings this reaction back this way, taking the phosphate off of this 2 position from fructose 2,6-bisphosphate. So you're decreasing levels of that. Remember, and so in turn, you're decreasing activity of PFK1 because remember, fructose 2,6-bisphosphate is an allosteric positive regulator of PFK1. So by shifting this reaction this way and decreasing these levels, you're decreasing activity of PFK1, decreasing flux of glycolysis. And then the other thing going on here is in the muscle, you're breaking down alanine, releasing it into the blood, and then alanine's making its way into the liver. And remember, alanine's being used in the state of fasting in the liver to synthesize pyruvate. The reason why you want to synthesize pyruvate is because that's the starting point of gluconeogenesis, which is the synthesis of glucose. You're synthesizing glucose from pyruvate. In turn, by increasing your levels of alanine, you inhibit pyruvate kinase. So you can see here, there's a few critical steps here where you're really inhibiting glycolysis all along the way. Really at the beginning here with glucokinase, so you're inhibiting there. Then you're inhibiting here at PFK1. Then here at glyceraldehyde 3-phosphate dehydrogenase. And then here at pyruvate kinase. Now, are you going to ask the question to list every enzyme that's being inhibited? Maybe. What I'm more pointing this out or counting these is just to give you further emphasize the point that this process of glycolysis in the liver is acted on at several key points of regulation. And you need to understand how this all tie together. And so just to close out this summary, the three ways that you're going to provide glucose for peripheral tissues from the liver is one, you're going to preserve glucose that's already present in the cell because you've continuously taking glucose up into the liver. The way you do that is by preventing glucokinase from phosphorylating glucose. So you preserve that glucose so that it can leave the cell. The other thing is you use your glycogen stores. So your glycogen stores, you don't want to have them end up going through the glycolysis process. That's the other reason, in addition to enhancing gluconeogenesis, the other reason you're decreasing the flux of glycolysis is to shift metabolism of glycogen away from glycolysis and towards producing free glucose, which can then again be released into the bloodstream. So that's number 2. And then number 3 is gluconeogenesis. And so these 3 processes are what helping you produce glucose to be used by peripheral tissues. And again, it all stems back to here, fatty acid oxidation, providing the energy for hepatocytes so that they don't need to rely on glucose for energy.
All right, so that closes out our discussion of the regulation of glycolysis. In the next lecture, we'll talk about how the regulation of glycolysis differs in certain scenarios and certain tissues.